Why does iron deficiency produce microcytic anemia? Many students memorize this fact, but understanding the mechanism makes it much easier to remember and answer exam questions. In this video, we'll follow the journey of a developing red blood cell to see exactly why iron deficiency leads to smaller red blood cells. Red blood cells are produced in the bone marrow from immature cells called erythroblasts. During their development, these cells divide several times while continuously synthesizing hemoglobin. Once enough hemoglobin has accumulated, the erythroblast stops dividing and matures into a normal-sized red blood cell. Iron is an essential component of heme, which combines with globin chains to form hemoglobin. Without sufficient iron, heme synthesis slows, leading to reduced hemoglobin production. This decrease in hemoglobin synthesis is the primary abnormality in iron deficiency. Here's the most important concept to remember. Developing erythroblasts do not stop dividing based on their size. Instead, they stop dividing only after they reach a critical intracellular concentration of hemoglobin. This is a favorite concept in medical school examinations. When iron is deficient, hemoglobin accumulates much more slowly. Because the critical hemoglobin concentration has not yet been reached, the erythroblast does not exit the cell cycle on time. Instead, it continues dividing for longer than normal. Each additional mitotic division produces two daughter cells. As the erythroblast undergoes more rounds of division than usual, the original cytoplasm is divided repeatedly among the daughter cells. Each new cell therefore becomes progressively smaller. This is the actual mechanism behind microcytosis. The red blood cells do not shrink after they are formed. Instead, they are born smaller because the precursor cells underwent extra rounds of cell division before maturation. At the same time, reduced hemoglobin synthesis means that each red blood cell contains less hemoglobin. Under the microscope, these cells appear paler, with increased central pallor. This appearance is called hypochromia. Together, iron deficiency produces microcytic hypochromic anemia. On the peripheral blood smear, you'll typically see small red blood cells, increased central pallor, variation in cell size known as anisocytosis, and in more severe cases, characteristic pencil cells. These are classic findings in iron deficiency anemia. The laboratory findings also reflect iron depletion. Ferritin, which represents iron stores, is decreased. Serum iron is low. Transferrin saturation is reduced, while total iron binding capacity increases as the body attempts to capture more iron. Mean corpuscular volume, or MCV, is reduced, and RDW is often elevated. Let's summarize the sequence. Iron deficiency reduces hemoglobin synthesis. Slower hemoglobin accumulation delays the signal for erythroblasts to stop dividing. The cells undergo extra mitotic divisions, producing smaller red blood cells with less hemoglobin. The final result is microcytic hypochromic anemia. This is one of the highest yield exam points. Iron deficiency does not cause mature red blood cells to shrink. Instead, it causes developing erythroblasts to undergo extra cell divisions before maturation because hemoglobin accumulates too slowly. If you remember this mechanism, you'll never have to memorize it again. Let's finish with one sentence that summarizes the entire concept. Iron deficiency slows hemoglobin synthesis, delaying erythroblast maturation. This results in additional mitotic divisions, producing smaller red blood cells with reduced hemoglobin content, the hallmark of microcytic hypochromic anemia. Thank you for watching, and happy studying! Follow us for more videos.